Hello everyone, we are Group 1 and today we'll be presenting on our study design of our skin microbiome mini project, which is about studying the effect of probiotic cream on the skin microbiome. These are what we will be covering in the presentation. Our skin microbiome is colonized by a large diversity of microbes, such as Staphylococcus epidermidis. These commensal microbes have a symbiotic relationship with us, producing antimicrobials that suppress opportunistic pathogens like Staphylococcus aureus. In addition, the skin microbiome plays a crucial role in modulating immune responses and supporting the skin's barrier functions. However, a dysbiosis in skin microbiome can lead to various skin conditions. For example, S. aureus-associated eczema and Cutibacterium acnes-linked acne. Moving on, we will be talking about probiotics. Probiotics, being common in products such as yogurts, are live bacteria that provide health benefits when consumed in adequate amounts. They support healthy microbiomes, digestive health, and immune function. The most commonly used genera in probiotics are Lactobacillus and Bifidobacterium, known for their role in maintaining a balanced microbiome and enhancing overall well-being. Recently, topical products claiming to improve the skin microbiome have become more common in the skincare market. However, there are very few studies that evaluate the efficacy of these products. Some probiotic creams contain preservatives to control the growth of microbes, but these preservatives can affect the probiotic strain viability. In addition, the probiotic creams do not have an abundance of probiotics in the product due to safety reasons. Instead, they utilize postbiotics to provide skin benefits, meaning these products no longer qualify as true probiotics since they lack live bacteria. Since the effects of probiotics on the skin are under-evaluated, we chose this topic in hopes to provide evidence-based recommendations for individuals looking for scientifically proven, probiotic-based solutions to enhance their skin health. Hence, we aim to investigate whether Rosken AD probiotic cream has any impact on the skin microbiome. The probiotic cream is stated to contain Streptococcus thermophilus, Lactobacillus bulgaricus, and colloidal oatmeal. Studies have shown that lactobacillus can enhance skin health as they regulate immune response and provide anti-inflammatory effects. Also, as thermophilus has been found to have anti-aging effects by promoting skin cell proliferation, stimulating collagen production, and inhibiting the breakdown of hyaluronic acid, an important component of the skin. In addition, colloidal oatmeal is a prebiotic that supports the growth of commensal bacteria like S. epidermidis and enhances production of lactic acid, which moisturizes the skin. Therefore, our research question is, does the application of Rosken AD probiotic cream lead to measurable change in the composition of the skin microbiome? We aim to find out whether this probiotic cream that claims to contain L. bulgaricus and S. thermophilus alters the skin microbiome. We believe that probiotics are unlikely to be alive in the cream based on our background research. Specifically for Rosken AD probiotic cream, it contains phenoxyethanol, a preservative that has a wide spectrum of antimicrobial activity, such as uncoupling oxidative phosphorylation from respiration and increasing permeability of cell membrane to potassium ions. Phenoxyethanol is effective against both gram-positive and gram-negative bacteria and is often used in bactericidal products. Hence, the probiotics in the cream might have been killed. Therefore, we hypothesized that we do not expect to see a rise in L. bulgaricus and S. thermophilus composition in the skin microbiome. In addition, we might also see significant changes in relative compositions of other microbes due to synergistic or antagonistic effects. Participants applied cream on the antecubital fossa of the left arm for 15 seconds twice per day, once in the morning and once at night. The cream was evenly spread in a circular motion to fully cover the antecubital fossa. The total duration of application was 10 days, where the first application was on the morning of 23rd of August and the final application was on the night of the 1st of September. The amount of cream applied was measured using one fingertip unit, which is the tip of the index finger to the first crease to ensure consistent dosing, as it is often been used to standardize topical treatments. Then, pre-treatment samples were collected on August 19th and kept in the fridge to preserve DNA integrity. Post-treatment samples were collected on the 2nd of September. Copan sterile swabs were dipped in saline solution and used to swab the antecubital fossa of the left arm for 45 seconds, and a total of 5 swabs were collected each time, and samples were labeled by their assigned barcode numbers. Some controls were implemented to ensure consistency in cream application and sample collection. For our intervention, the dose, motion, and duration were standardized, and nighttime application was only done after showering. Post-treatment samples were collected approximately 12 hours after the last cream application and before showering in the morning. To ensure compliance and consistency, we recorded the times of last cream application and sample collection. We chose this 12-hour interval to give enough time for the cream to be absorbed into the skin and exert its effects on the microbiome. Standardization also ensures that any differences in time intervals between the participants does not affect our results. Next, we chose the anticubital fossa because it is a moist skin site that sweats more and can promote the growth of certain microbes. Skin commensals like S. epidermidis can grow better with more sweat by using urea as a nitrogen source. Hence, we hope to attain a higher microbial load by sampling the anticubital fossa, which other microbiome studies have also done. Lastly, doing 5 swabs for each sample collection allows us to pool the DNA for a higher microbial load. Cotton swabbing is preferred to scraping because it captures a higher variety and number of microbes, while also giving higher precision and accuracy of identification across different microbial classification in microbiome analysis of 16S sequencing results. However, we could not control for equal intensity, speed, and angle of swabbing across participants. This may result in some rougher participants collecting more dead skin cells, which contaminates the DNA concentration quantified by the nanodrop. If we can redo our study, we will choose to limit showering frequency each day because it can remove the cream and reduce its efficacy at changing the skin microbiome. To ensure consistency, we can have the same person to perform swabbing on everyone. We can also collect swabs from both anticubital fossa, which are treated with cream, to capture higher microbial load. There are 4 limitations to our study. Firstly, the probiotic cream was only applied twice a day for 10 days. This might be too short to record reliable and conclusive results, as microbiome might not have stabilized. This affects the evaluation of the cream's effectiveness. A remedial measure would be to increase the duration to 14 days to match clinical studies investigating the effects of topical cream to skin microbiomes. Sampling should also be conducted at various time points to monitor the changes in skin microbiome in real time. This allows us to capture both transient and longer-term changes to the skin microbiome. Secondly, our sample size is too small to be significant, with only 4 pre-intervention and 6 post-intervention results. The sample size will provide low statistical power and is too small to achieve statistical significance. Moreover, this limited sample size increases the risk that individual differences will significantly influence the results, making it challenging to draw reliable conclusions. Additionally, the small sample size is not representative of the general population. Thus, the findings might have low generalizability to the wider population. To enhance the reliability of our results, we can increase the sample size of our study to at least 14 participants, as done by other clinical studies with similar objectives. Thirdly, lifestyle and environmental factors affect the skin microbiome in various ways. Diet influences the gut microbiome, which in turn affects the skin microbiome via the gut-skin axis. Exercise can also influence the skin microbiome, increasing the diversity of microbes. Lastly, temperature and humidity of one's living environment also affect the skin microbiome. Therefore, these are important confounding factors which should be taken into consideration. These factors should be standardized in an ideal study where participants live, eat, and exercise together to eliminate such confounders. Alternatively, information on diet, exercise, and living environments should be collected to group the participants. Lastly, within our group, there are some individuals with pre-existing skin conditions, such as eczema, while others do not have any skin conditions. This difference can lead to variations in the existing skin microbiome, which can affect the response to the probiotic cream. This variability presents a challenge for generalizing our results between diseased and healthy populations. To remediate this limitation, we recommend collecting information about participants' existing skin conditions. This will allow us to gain a better understanding about the potential confounders and how they can affect the results of the study. Alternatively, we can categorize participants into 2 distinct groups, one comprising participants with skin disorders, while the other group contains healthy participants. This approach allows for a clearer understanding of how the probiotic cream affects different skin microbiomes, improving the validity and applicability of our findings. Here are our references. Thank you.